Thank you. Totally forgot. Okay, so again, we're on the red, targeting the plasma membrane. The overall goal, poke holes in the membrane, okay? So we've got two drugs here that are targeting the plasma membrane, polymyxin and daptomycin. So I'm gonna write on that without talking, so hopefully it doesn't get mad at me. Yeah, exactly, Shelby. See, I'm big with alliteration and things like that to remember. So yeah, polymyxins poke the plasma membrane. Good. So remember, polymyxins specifically are going to target gram-negative cells. I'm going to put that in the table. Remember, polymyxins do that because they have an affinity for lipopolysaccharide, LPS, which is only found in there you go, yep, which is only found in the outer membrane of gram negatives, okay? So polymyxin is going to make a leaky membrane for gram negatives. Daptomycin is more for gram positives because it doesn't have an affinity for anything except the plasma membrane. And so remember, for gram positives, they don't have an outer membrane, okay? So all they have is the plasma membrane for these drugs to target. We tend to use daptomycin for gram positives. And again, I'm going to kind of write off the side goal here. Leaky membrane, poke holes in it. All right, is this any better? Okay. I'm sorry, y'all. I know that's got to be frustrating. I can't figure out why. I'm literally not even moving my head anymore. I'm just in the exact same spot talking. Okay, so for the green, moving clockwise, this is going to be nucleic acid synthesis. So I'm going to abbreviate that in green as Na synthesis, just for space. I'm going to go ahead and fill this in, and then I'll talk it through just to try to minimize pissing off Blackboard.
What did you say they target again?
Nucleic acid synthesis, so making DNA or RNA. All right, so I just put one word for each of these, but I'm going to elaborate. So when it comes to nucleic acid synthesis, remember, we have two types of nucleic acids, DNA and RNA. So you may want to make a little note where I wrote on the bottom green one, the quinolones, also the fluoroquinolones, those are going to be targeting DNA replication. Okay, so DNA is the target for the quinolones and the fluoroquinolones. And they target DNA replication by binding to topoisomerase. So remember, topoisomerase was responsible for relieving supercoiling during DNA replication. That prevents DNA from getting wound up too tight and breaking. So by blocking or by binding to topoisomerase so that it can't do its job, DNA gets wound up really tight, it breaks, and then the bacteria can't survive. So that's what the quinolones and fluoroquinolones do. They target DNA replication. So I would write that on there somewhere just to make sure you remember what topoisomerase does. And then, yes, exactly. When it comes to rifamycin, that's the one that starts with an R, and it's targeting RNA synthesis. Now, you might want to make a note. That's also called transcription. I'd love to bring in Unit 1 stuff when I can. So the process of making RNA is transcription. The drug is rifamycin, and it binds to RNA polymerase. By binding to RNA polymerase, it can't bind to the DNA, which it needs to make RNA. Because remember, y'all, transcription, we start with DNA, we make RNA. But if the key player, RNA polymerase, can't access that DNA, then it can't make any RNA. Are we feeling okay with these? Is this helping at all? Sometimes I wonder if these graphics only make sense in my mind. Now, please, I'm not offended. If it doesn't, I know not everything is going to work for every person. So if it doesn't work for you, please know I'm not going to be offended. For the next one, we're going to be building our own. I want to kind of help teach y'all how to make these yourself, because it's one of those things that took me a long time to figure out. Okay, so let's keep going. Purple. Purple obviously is the biggest one. So this is translation. I'm going to stop talking and kind of fill in the flow chart, but I'm only going to put keywords, okay? So you may want to wait until I babble on about it if you want to put more information. So give me just 1 second while I do this. I ran out of space for that one. That last one is chloramphenicol. I'm going to go ahead and write my keywords in there. 1 second. Okay. Sorry, it looks like chicken scratch, but let's talk through it. So we're going to start on the left side, the light purple. So let's make a note so that in general, purple is translation, a protein synthesis. So I'm going to write TSL for abbreviating. So this is translation, protein synthesis. Now remember, I did 2 shades of purple because remember, with translation, we have 3 key players: the messenger RNA, the transfer RNA, and the ribosome. All of these antimicrobials directly bind the ribosome. But remember, the ribosome is made up of the large subunit and the small subunit. So these 2 drugs on the light purple, that's the small subunit. So I'm going to write that in where it branches off into the light purple. I'm just going to put small. And then for dark purple is going to be the large subunit. We have 3 of those. All right. So tetracycline and aminoglycosides, they both are going to target the small subunit. Tetracycline binds the small subunit in such a way that incoming transfer RNAs can't bind. Now that's a problem because transfer RNAs are what deliver the amino acids. So if transfer RNAs can't get to the ribosome, they can't drop off amino acids, so the protein can't be built. Huge problemo. Okay. So for tetracyclines, tRNAs cannot bind, so they cannot drop off the amino acids. Is that better? Okay, thanks for y'all's patience. I know this probably is annoying for y'all as it is for me. Come on, Blackboard, get it together. All right, aminoglycosides. These also bind to the small subunit. And if you look at the picture, this is where pictures are very helpful. With the aminoglycosides, they bind right where the messenger RNA is kind of laid down, but it does so in kind of a bumpy fashion. Okay, so if you take a look at that picture later, you'll notice that the aminoglycosides don't bind in a straight line. And so what it does is it causes the messenger RNA to kind of be like wavy. And so it causes the messenger RNA to be misread. Because remember, y'all, we read the messenger RNA in sets of 3. But normally it's a straight line. So it's very easy to go, okay, these 3, now these 3, now these 3. But when the messenger RNA gets kind of wavy, it's hard to know, well, is that number 3 or is that number 2? Did I do that one? Ugh, I don't know. And so it kind of gets all cuckoo birds, for lack of a better term. So it causes misreading of the messenger RNA. Now for the 3 that target the large subunit, we're going to start with the oxazolidinones. And again, this is where I think pictures are fantastic. With oxazolidinones, the drug binds at just the right point so that large subunit cannot attach to the small one. So in other words, the ribosome can't actually form. The 2 individual pieces of the ribosome cannot get together, which means translation can't even start. So this is really blocking the initiation of translation. So the key players cannot get together, so translation cannot start. All right, I'm going to power through these next 2 and then I'll stop for questions. So if you have them, be putting them in the chat. All right, erythromycin. So with this one, I need you to try to visualize translation, right? That's easy. Not. So we have our ribosome completely intact, and it's got the messenger RNA kind of threaded through it. Once the ribosome reads one codon, it attaches the amino acid, and then it scoots down the messenger RNA, and it reads the second codon, attaches that amino acid. Then the ribosome moves again, reads the next set of 3, attaches the amino acid. Ribosome moves down, reads the next set of 3, attaches the amino acid. That movement of the ribosome is called translocation, just a fancy word for move. And so erythromycin binds in such a way that the ribosome can't move. It can't continue reading that messenger RNA. So this is really targeting elongation, the elongation step of translation. All right, the last one for chloramphenicol. This one's really, they're all very effective, but chloramphenicol is pretty potent. And what it does, again, if you look at the picture, it's going to bind the large subunit right in between where the transfer RNAs attach. Because remember, transfer RNAs are dropping off amino acids. Those amino acids have to be attached to one another, right, to build a protein. So chloramphenicol binds in such a way that the amino acids aren't connected. They're connected through peptide bonds. So we say that chloramphenicol prevents peptide bond formation. Prevents peptide bond formation. That just means the amino acids aren't attached together, so proteins aren't actually built. And of course, without proteins, any living thing would die. All right, y'all, how are we doing with this? I know it's a lot. Do we have any questions on things we've said? Can y'all still actually hear me?
For tetracycline, how does it prevent the tRNA to go into it if it's binding to the small subunit?
Because it binds kind of like at the surface, at the tippity top of the small subunit. So think of it as kind of like Legos. Normally the large subunit kind of attaches, links on to the small subunit, but with the tetracycline there, it acts as a bumper. It prevents the large subunit from actually latching on because it blocks those binding sites. Awesome. Yeah, if y'all do have helpful phrases, like share away. Some of them are easier than others, right? I'll see if I can think of any. It's kind of hard with all these crazy words like oxazolidinone. Like what? Who named these? All right, last one, y'all. So remember this last one, these are the sulfa drugs, okay? Sulfa drugs. And these are going to target folic acid synthesis. Okay, so I'm going to fill this one out and then we'll talk about it. Okay, so bacteria are different than us, well, in a lot of ways, but for this conversation, bacteria make their own folic acid. Okay, folic acid is a really important precursor molecule for things like making DNA, making RNA, making, what did I already say? Proteins, DNA, RNA, all the things. Okay, folic acid's really important for that. Bacteria make their own folic acid. We do not. We get our folic acid from our diet. So this is a great drug for us to take because we don't have the pathway that's being targeted. Remember, it's that whole idea of selective toxicity. So basically, these sulfa drugs do competitive inhibition. So notice, y'all, that's 2 drugs that do that do competitive inhibition. Okay? And so the sulfa drugs mimic the shape of the substrate that's used to make folic acid. And so the sulfa drugs get into that active site of the enzyme, shut it down, and so folic acid can't be made. And if the bacteria can't make folic acid, they can't make DNA, they can't make RNA, they die. All right, y'all, questions about this? So for some of the drugs, I don't have anything super clever off the top of my head, but like tetracycline deals with tRNAs. So the tetracyclines block block tRNA. And I think in one of my classes, we were talking about the aminoglycosides, the one at the top of the left-hand column. If it misreads the messenger RNA, it messes up the amino acids. So kind of using the name aminoglycoside as a hint. But yeah, there's some good ones in here. Okay, questions on this? I want to make sure we get to some other stuff. All right, if you think of one, let me know. Let's go ahead and move on to the microbial growth flowchart. Oops, that's the answers. Y'all can't see that. Where's it at? Let's see. Now I can't find the blank one. Is this it? All right. So I've had a lot of students actually mention lately that, right, flowcharts are really helpful, but they can be really hard and intimidating to make, right? Now, in lab, y'all are going to be practicing this. But what you kind of have to do is you're just mapping out the lecture. So I know you may not have your notes in front of you, but if you go back and look at them afterwards, I think you'll see kind of how this map, which probably looks crazy right now, is actually connecting some of these dots. So let me get mine set up to draw. And let me make sure I can still see the chat. So 1 second, y'all. Okay. So, and now this is normally easier to make your own than try to figure out how somebody else was thinking. especially me. My in-person kind of a hot mess at the time. All right, so if you think about our conversation on microbial growth and what microbes need in order to grow, we divided those needs into two categories. Okay, does anybody remember what we talked about? Yes, there we go. Yep, chemical and physical factors. Okay, so how I did it, I put the physical factors right here and chemical down here. I built out the top a lot more than the bottom. So remember, when we think about what microbes need to survive, their actual physical environment is really important. So that's what the physical means up there. I'm going to put environment. I don't know why this is blue. Hold on. Okay. So we've got our physical environment. And then remember, y'all, when we talk about chemical factors, we're talking about like nutrients. Okay, so you might want to make a note of that. I really need to invest in a tablet. This writing on a computer is crazy. All right, so when we think about the physical environment, we broke that big category down into 4 smaller categories. So I'm going to go ahead and fill it out and then follow along. We'll talk through it. So within physical environment, we have to consider the temperature of the environment, the pH that the microbes need, tonicity, and then oxygen. Now remember, y'all, when I say oxygen, I'm talking about like atmospheric oxygen, like O2. Okay, so thinking about temperature, we classify bacteria based on the optimal temperature that they need to grow. Okay, so on your own, think about those 4 classifications of bacteria, and that's what's going to go in these 4 boxes. Okay, and if you want to even test your brain even more, write the name of the group and then also include the optimal temperature. Because remember, you're going to have to know that for the exam. All right. I know you probably need more time, so I'm sorry to rush you. What was the group of organisms that like the cold? What were they? Exactly, the psychos. Yes, psychrophiles. So I'm going to abbreviate just because the boxes aren't very big for me. And does anybody remember, what is their optimal temperature? Good, so yeah. And if you want to, if you are very linear, you can even draw another line like this, have a little box, and then you just say 10 degrees Celsius. Or you can just put it in parentheses, however your brain works. All right, what's my next group and what's their optimal temperature? And feel free to abbreviate. Good, yes, the mesophiles, and that's at our body temperature, y'all. So pathogens, human pathogens, normally fall into this category because they grow best at our body temperature. So we make great habitats for them. All right, who's next? Good. Thermophiles around that 67 to 68. And then finally, we'll have the hyperthermophiles. So y'all go ahead and tell me what temperature they are, since I accidentally said their name. All right, so it's how I'm, exactly, good, Stormy, actually reminded me, I almost forgot to say that. Yeah, so the thermophiles, thermo means heat. These things love the heat, okay? There's gonna be some wiggle room. So it's normally on the graph, kind of how you're looking at it, between 68 to 70 degrees is normally fine. I won't be that kind of nitpicky on the exam. And then our hyperthermophiles, hyper, right, means like really love. So hyperthermophiles really love the heat. Oops, sorry, throwing things. All right, so is it kind of making sense how I'm mapping this out? So microbial growth, big picture, physical environment, and nutrients. Then you're kind of zooming in. Okay, within the physical environment, what things do we have to think about? And then zooming in. Okay, let's just talk about temperature. We've got these 4 categories. Okay, so then y'all tell me, what do you think go in those 3 boxes for pH? Exactly. And now remember, we're going to be using terms for classifying them. So exactly, acidophile, neutrophile, alkalinophile. Exactly. And then go ahead and put their optimal pHs as well. Now, I am going to be more like dramatic on exams. I'm not going to say, an organism grows at a pH of 6 and make you be like, is it neutrophile or acidophile? I'll pick something like a pH of 2, a pH of 12, okay? It's an alkalinophile. Yep, and so exactly. So remember, y'all, acidophiles love acidic pHs, but you have to remember chemistry that acidic pHs are low pHs. Because I could say to you on an exam, you discover a bacteria growing at 68 degrees Celsius and a pH of 2. How would you classify it? You would have to go, okay, pH of 2, that's low, so it's acidic, so the organism's an acidophile. And then same thing with temperature, okay? So kind of get ready for some little scenarios kind of like that, where I describe the environment and you tell me how to classify it. All right, so for tonicity here, we were talking about creating a hypertonic environment. So remember, that's like storing food in really salty environments, really sugary environments. Because remember, in a hypertonic solution, plasmolysis occurs. And that's basically where all the water is rushing out of the cell is going to die. But we talked about some organisms that love salt, because that's what we're going to talk more about in lab and in Unit 4, some human skin diseases. And the reason they happen is because the organisms actually love salt. So exactly. So we're talking about halophiles and haloduric. So it is, I only put one box here because we typically will talk about halophiles, but you're absolutely right, Storm. Halophile and haloduric. So you can put both of them in that box or add an extra box. But what you really need to know is when you see halo, we're talking about salt-loving organisms, which are going to be the staph. If you started, working on lab stuff for like mannitol salt agar, right? We talk about how the staphylococci love salt. They live on our skin. They love it when we sweat because they can just chow down on it. Isn't that a great image? All right, so now for oxygen, I think you're starting to figure out how this mapping goes. Go ahead and take a minute on your own and kind of fill out those boxes. And it doesn't matter which order you put it in. I would recommend put a little definition or keyword next to it to make sure you remember. So not only do you need to recall the 5 different classifications, you of course need to know what makes something this versus that. So I'm going to start doing it and then we'll talk about it. All right, just for the sake of time, I'm going to go ahead and start talking. But I do recommend y'all, if this is helpful for you, like presenting information this way, print this out, and before the exam, practice filling it in. Make sure you know all these terms, you know their definitions, because if you can do that, this exam won't be too tricky for you. All right, so I like to think on terms of like a spectrum. So that's why I listed mine this way. So I'm going to go ahead and go through them real quick. So the first one is an aerobe. You can also call it an obligate aerobe. But normally if you say an organism is an aerobe or aerobic, you assume the obligate part. So these are the ones that must have oxygen. Without it, they die. Microaerophiles. So again, break down these words, y'all. So see where it says aerophile? That means it loves, phile means to love. It loves air, but the micro means small. So it loves small amounts of air. So what that means is normal atmospheric oxygen, like we breathe, is too much for these organisms. They need a little bit less than what's in the atmosphere. So you tend to see these organisms right at like the surface of water, like right below the surface of water. So they're not quite getting atmospheric oxygen, they're getting just a little bit less. So microaerophiles need oxygen, but smaller amounts than what's in the atmosphere. I'm going to save the next two for last because that's what people tend to get confused. Hence, so you'll probably see it on your test. So an anaerobe or an obligate anaerobe means they cannot handle oxygen. They can't tolerate it whatsoever. And remember, this is normally because they don't have catalase. They don't have peroxidase. They don't have superoxide dismutase. Those enzymes that were listed on that slide, because remember, those enzymes take harmful forms of oxygen and convert them into something safe like water. Well, if an organism doesn't have those enzymes, then it can't get rid of the harmful oxygen, and the harmful oxygen will kill it, okay? All right, so that leaves us our facultative anaerobes and our aerotolerant anaerobes. So again, let's use our names to help us remember what they do. Aerotolerant. It's called that because it literally tolerates air. It cannot grow in the presence of oxygen, but it's not going to die. So think of it, I don't really have a great example. So, if we go underwater, again, please note, not my best example. We can hold our breath, right? And so we can't actively breathe underwater, but we can tolerate being without that oxygen for a little bit. All right, so that's what these aerotolerants do. When oxygen is around, they can technically just chill, they can exist, but they can't actively divide and do normal cell stuff, okay? So with aerotolerant, they cannot use the oxygen. They cannot grow in the presence of oxygen, but they can still survive. They literally tolerate it, okay? For facultative anaerobes, these are the switchers. They don't care if there's oxygen or no oxygen. They are fine either way. They can do aerobic respiration. They can do anaerobic respiration. So you change the environment, they don't even care, okay? They grow better with oxygen, but they can grow either way. Okay, so the facultative anaerobes are the switchers. They can grow with or without oxygen, but they do grow better when oxygen is present. All right, how are we feeling about this? The content and the presentation of it. We doing okay? All right. So I didn't build out the bottom quite as much. Honestly, there just wasn't quite as much to put. So remember, with the chemical factors, the nutrients, we have our three categories. We have our macronutrients, which are the ones that the bacteria need in large amounts. We have the trace elements, which are needed in very little amounts, and then vitamins. So that's what goes in these boxes. Hold on. All right, so our macronutrients, vitamins, which are often gotten from the environment, they help enzymes and things work better. The trace elements are things like copper and zinc, little ions that certain processes need to happen. The ones I'm more concerned with you knowing are the macronutrients. So had I had more space to build this out, I would come over here like this. I'm going to kind of do it now. That's nitrogen. Okay, so carbon, hydrogen, oxygen, nitrogen, sulfur, phosphate. Those are the macronutrients. Now, what we're going to do tomorrow is we're going to talk about why we need, well, we need and bacteria need. What are they used for? So on your own, after the review, if you want, come over here and go, you know, phosphate. Okay, this is for phospholipids. It's for nucleic acids, because remember, nucleotides include a phosphate group. So these are nucleic acids, and even ATP, right? Adenosine triphosphate. So you could build this out to where you're saying what each of these elements is needed for, but we're going to do that tomorrow, so I'm not going to spend any time here. Do we have any questions about this? Any of the terms, anything at all? All right, so then what we're going to do last, and we may not get all the way through it. I'm happy to stay a few minutes late. I know with the sound difficulties, it kind of slowed us down at first. Is this lovely, so easy to follow, respiration review sheet. Okay, now notice there's a legend, so it kind of gives you some guidance, but yeah, Shelby, please don't be alarmed if you're looking at this going, what the crap? That's what I'm here for, okay? So, but it is a nice way to kind of summarize it. Once it's filled out, you're going to be like, oh, all right. So don't worry. Here we go. And again, if you do need to leave right at 6, please do what you need to do. It's being recorded. But I am going to just go ahead and go through this worksheet. All right. So I want y'all to think about at the beginning of aerobic respiration, what do we start with? What is our starting molecule? Good, glucose. It's a 6-carbon molecule. That's what those 6 circles mean. 6-carbon molecules. This is glucose. All right, what's the first step of respiration? There we go, glycolysis. So remember, y'all, I'm going to kind of give you a little bit more information, just so we can kind of be doing. Remember, glycolysis literally translates into sugar splitting. Glyco means sugar, lysis is to break or to split. So with glycolysis, we're going to split glucose into 2 molecules of pyruvate. And so that's what the little number question mark means. It's trying to say how many of that molecule. So I'm just going to write it on the line. So notice in the legend, the little kapow symbol, that's ATPs. So wherever you see that little kapow, you're going to put ATP. And so we're going to have 2 of them. Okay, so again, I'm sorry I can't write very pretty on this. So 2 ATPs, 2 pyruvates. What else is made during glycolysis? What am I missing? What's my stored? There we go, good. 2 NADH. Remember, that's the stored energy. All right, now you might remember I told you that during glycolysis, I used the stocks example during class. If you want to get money back from a stock, what do you first have to do? Invest money, right? So that's what this top little kapow symbol is for. So in order to get respiration started, the cell has to invest 2 ATPs, right? So during glycolysis, technically 4 ATPs are made, but because we have to pay off that debt of 2, we take away 2, okay? So with glycolysis, we convert 1 molecule of glucose into 2 molecules of pyruvate. We also get 2 ATPs, which is usable energy, and we also get 2 forms of stored energy, which is NADH, okay? So looking at the little ATPs that we just made, notice it says how. Remember, this ATP is made in 2 different ways: substrate level phosphorylation and oxidative phosphorylation. For glycolysis, it's made with substrate level phosphorylation, which means that it just stole a molecule, or I'm sorry, it stole a phosphate group from some other molecule. And we're going to review some of these a little bit more on Saturday. Okay, so remember, the next step is going to be the linking step, which is called pyruvate oxidation. And remember, oxidize, that's the science word for break down. We need to continue to break down this pyruvate so we can get as much energy as possible, okay? So pyruvate oxidation is next. That's the linking step. And so remember, for every pyruvate that we start with, we make 1 acetyl-CoA, okay? But because we have 2 pyruvates, we're going to have 2 acetyl-CoAs. That's what ACA stands for, is acetyl-CoA. We're also going to get 2 molecules of stored energy, which is NADH. And then something that I will not put on your exam is carbon dioxide. I only worry about energy stuff, so don't worry about these little circles here. It's just carbon dioxide, but I don't care about that. We're worried about energy. Okay, so, oh, I see what they're doing here. Okay, so for this chart, and you don't have to know this, but I'm going to explain it so that you don't get confused later. Pyruvate gets converted into the molecule acetyl, and then this is the CoA, and so it combines to form acetyl-CoA. That's an unnecessary detail. So you just need to know that the linking step makes acetyl-CoA. And so this linking step is important because it's linking glycolysis to the Krebs cycle. Because the starting molecule of the Krebs cycle is acetyl-CoA. Now remember, only 1 acetyl-CoA can go into the Krebs cycle at a time, okay? So when 1 acetyl-CoA goes in, we get 1 ATP, 1 FADH2, and 3 NADH. But notice on this graph, it says 2 times because we have 2 acetyl-CoAs. So the first one goes in, makes a bunch of energy, then the second one goes in, makes even more energy. So we're going to put the net total here. So that's going to be 2 ATPs. And again, that's through substrate level phosphorylation. They stole a phosphate group. We're going to get 2 FADH2. And then 6 NADH. All right, so at this point, all we have left are energy, right? We've got usable ATP, we've got stored energy, and that's it. So there's nothing left to break down. Now we need to deal with all that stored energy, right? Because the cell can't use NADH and FADH2 as is. It can only use ATP. So we need to take all of that stored energy, and if y'all look at this picture, and it's hard for me to show you with one screen, do you notice wherever there's NADH, you see how it's all cycling down here to this last step? You see how that happens? That's because, remember, step 4 is oxidative phosphorylation, and I do not have room to write that. But remember, oxidative phosphorylation has two parts: the electron transport chain and chemiosmosis. Chemiosmosis. So NADH and FADH2, they all drop their electrons off at the electron transport chain. So that's why all those arrows go to step 4. So if you remember, and if we have time tomorrow, I'll go over this last step in more detail. But remember, NADH and FADH2 drop off their electrons, and they kind of play hot potato with them, right? And so at the end of the day, oxygen, this is why it's aerobic, oxygen says, fine, I will take these silly electrons. That's what goes right here. The final electron acceptor is oxygen. This is why we need oxygen for this process. When oxygen accepts those electrons, it becomes water. And then the whole point of this was to make lots and lots of ATP, 26 to 28 molecules of ATP. Okay, so it looks scary. There's a lot happening, but hopefully now that it's done, you kind of see like, oh yeah, I did know this, just presented in a slightly different way. Are there any questions about this, y'all? All right, well then that is it for today. Thank you all for coming. As you study over the weekend, remember, if you need to get a hold of me, Remind app is going to be better. We're trying to get this house in order. We're going house hunting. And so I just won't be at my computer as much. So please message me on the Remind app if you need anything. Y'all have a fabulous night, and hopefully I'll see y'all on Saturday. Y'all are welcome. Have a good night.